There he is. Thank you.
All right, talk about pressure. Thanks, Dr. Silver. It's a great pleasure to be here today to speak with you. My laboratory studies a variety of heme malignancies. We're interested in acute myeloid leukemia, interested in the genetics of that disorder. We also study development of novel therapeutics for AML. But I'm also, in the past few years, have become interested in the myeloproliferative neoplasms, particularly interested in myelofibrosis. And that stems from my laboratory's basic interest in development of cells called megakaryocytes, which I'm sure you've heard of. These are cells that give rise to platelets, and they play critical roles in the development of the MPNs, particularly essential thrombocythemia, where you have too many megakaryocytes and too many platelets, but also in myelofibrosis, where we think that the abnormal megakaryocytes directly contribute to fibrosis. So I've given lectures to a few patient crowds. That's in my role as a scientific advisor for the Myeloproliferative Neoplasm Foundation. And I've given a lot of lectures to medical students and to undergraduates. So I'm trying hard today to give a lecture to you that you'll understand. And I certainly welcome questions at the end of the session or at lunchtime or any of the breaks. I think it's important to understand at least parts of the basic science behind these diseases because it gives you insights into how we're going to try to develop new therapies for these disorders. So let me begin. Let's see if I can make this work properly. And it's going to be a little tough because there's two projection screens. And so when I show you, I'm going to have to use a laser pointer on one, but I'll try to use it on both sides.
We can get you two laser pointers.
That's right. I'm a little dyslexic, though. So the agenda for this morning, this brief talk, will be threefold. First, to review mutations. And so many of you have heard about mutations that you may have in your blast or in your hematopoietic cells. These are changes in the DNA that affect gene regulation or function of particular important proteins in your body. And of course, I'm sure you've heard of JAK2 mutations. I'll give you a quick overview about what JAK2 mutations are doing in your body. And also, you may have heard of a gene called MPL or MPL. Those mutations are fairly common also in ET and PMF. And then I'll tell you about some other mutations that you may or may not have heard of. These are terms that may be thrown around, may be mentioned by a clinician, or will be discussed later today. And I'll try to give you an understanding of what those mutations are doing to your cells. Second, I'll talk about one of my favorite topics, are stem cells. And there's a lot of, I think, misguided information out in the public about what are stem cells. What do they do? Why are they important? So to give you, I think it's fun just to give a very basic background to stem cells, what they do. And then we'll talk about what are called leukemia stem cells or tumor-initiating stem cells. These are the cells that we think give rise to persistence of disease. And these are ones that are very difficult to target. So even though we may have drugs that target the maturing cells or may help you get some therapy, we really need to target these, what we call tumor-initiating stem cells, as a way to develop better therapies. And then finally, I'll talk about fibrosis, just a very simple introduction about what we think fibrosis is, how do you understand it, and then talk a little bit about the mechanism of fibrosis development. And I think this is important because one of the next steps we need is to figure out how to cure or reverse fibrosis that occurs in the bone marrow. Some of the slides are a little complex, but I'm going to take you through them, and I apologize if they're not large enough to see in the audience. But this is, so the take-home message from this slide is that MPN cells harbor mutations commonly in JAK2, but also can have mutations in MPL, MPL. And this is a schema, this is from a review of Ross Levine, who's one of the leading clinical researchers in the field. And I'll just focus first on the bottom panel. So we know that polycythemia vera is characterized by a high number of red blood cells. This disease is characterized by having a very high percentage of JAK2 mutations. So more than 95% of patients will have a JAK2 mutation. They can either be the common JAK2 V617F mutation, which I'm sure you've heard of. They can also be a different mutation in what we call exon 12. Actually, I'll give you a quick discussion of what an exon is in a few minutes. These mutations alter the function of JAK2, and I'll show you that on the next slide. Essential thrombocythemia, where we have too many platelets, often have mutations in JAK2 also, but not to the same degree as you see in PV. So about 40 to 60 to 70% of cases will have a JAK2 V617F mutation. And there's also mutations in MPL in about 5 to 10% of cases. And I'll explain what MPL is on the next slide. And then finally, in primary myelofibrosis, we see about a similar incidence of mutations. The MPL gene is mutated in about 10 to 15% of cases, and the JAK2 is mutated in, say, 50% of cases. Why I'm highlighting this is that we don't quite know now what are the other mutations that are driving ET and PV. If you don't have a JAK2 mutation, how do you have accelerated growth of these cells? How are you causing the disease? And that's an intense area of research that I'll also focus on in a few minutes. Also on this slide, it's a little more complex because we don't know what else you need, or we're beginning to understand what else is needed besides a JAK2 mutation. And so what's illustrated here is this is one of these stem cells I'll mention, the hematopoietic stem cell. And this is the cell that gives rise to all the blood cells in your body. And there's some suggestion that these cells may have what we call an inherited predisposition allele. So it might be the case that in some families there's a mutation that will make you more susceptible to getting a JAK2 mutation and then having disease. There's also what we think to be downstream of that, something called a pre-JAK2 allele. An allele means, you can think about allele as one copy of DNA or a piece of DNA or a gene that has a mutation or not. And so this pre-JAK2 allele could be something else that's mutated that then cooperates with JAK2 to cause disease. And then finally, the other point on this slide is something called genetic modifiers. So this would be other changes in your DNA that might also participate. And so just want to underscore the complexity of this disease. We all think about JAK2 as being the most crucial driver of the disease, and that may or may not be true. It's certainly the case that we need other mutations to cooperate with that to cause disease. So on this slide, I'm going to take you through it kind of slow, but what is this showing? This is going to explain to you what a JAK2 mutation is doing. And before I tell you, I'm going to try to give you an anecdote that I just came up with, which I hope makes some sense to you. Let's say you go to a restaurant, and the waiter comes up to you and asks you what you like to eat. And you give them you want to have a steak, you want to have an appetizer, and you're going to have a dessert. This, what we call this in our parlance, a signal. This will then be, your order will be then taken into the kitchen. It'll be told to multiple people on the kitchen staff, and these people will respond. They'll make your steak, they'll cook your steak, they'll cook your appetizer, and then they'll also make your dessert. And so just by you entering the restaurant, you set up a process that has an end result where there's multiple events that are happening. Okay? So that's how JAK2 normally works. And so on the left part of this, in A, what we have here is a cell. And on the outside of the cell, we have what are called receptors. These are things that bind what are called cytokines. And cytokines are secreted factors that just float around in your blood. Okay? So one of these cytokines is what we know as erythropoietin. You may have heard of that. That's a protein or a cytokine that makes red blood cells. The way that erythropoietin works is to bind to this receptor, and the receptor is, in this case, the EPO receptor. It can also be what's called the MPL, is also a receptor. So what happens when this protein, this cytokine, binds to the receptor? Well, it gives rise to a signal. It's analogous to you entering a restaurant and having a seat. It leads to these other processes. And so what does it do? Well, when you bind, you activate JAK2. Normally, JAK2 doesn't do anything. So if you don't go to the restaurant, they're not going to cook the meal that you want. So when JAK2 is then activated, it activates multiple pathways. Again, just like your order activates 3 different cooks. And so JAK2 then can signal through multiple pathways. And so each of these are what we call pathways. The one that you may have heard of is called the JAK-STAT pathway. So JAK activates these proteins called STAT. What do STATs do when they activate? Well, they move into the nucleus of the cell. That's kind of the engine of the cell, let's say. It's the region of the cell that makes important decisions. So when STAT goes into the cell, it communicates something. So again, you can think of STATs as the waiter entering the kitchen, telling them what to do. So you need this STAT signal in order for the cells to grow in response to the cytokine. There's other pathways, though, that are also important. There's activation of what's called the mTOR or PI3 kinase pathway. And you don't have to understand, you don't have to know that the specifics of this won't be on the exam. But there's another pathway called the RAS-ERK kinase signaling pathway. Okay, so what's the point of all this? Well, the point is that this tells you there's multiple targets for therapeutic intervention. We think that if the JAK inhibitors are trying to block the activity of JAK to turn on all these pathways, and primarily we think about blocking this STAT protein. But there's other pathways that we could potentially target, including this PI3 kinase mTOR AKT pathway. We can imagine if we develop drugs to that pathway, they might have some efficacy. And in particular, if you combine those with JAK inhibitors, you might have even better ability to stop the disease. Okay, so what happens when you have a JAK2 mutation? That's shown in panel B. We no longer need this signal, so we don't need someone to come into the restaurant. Instead, they're going to make your meal anyway, okay, which we know is not, it's kind of a waste, right? And so how does that work? Well, JAK2, when it has this V617F mutation, it doesn't need the cytokine anymore. It just works all the time. And so what I mean by working is that it activates all of these signal pathways. It turns on JAK-STAT, turns on this PI3 kinase pathway, and also turns on this ERK pathway. And we think that you need all of these events in order to get the disease to occur. And then finally in panel C, this is the MPL mutation. So MPL is another one of these receptors. It binds on a cell membrane, and it normally interacts with a protein called thrombopoietin. And these are important in making megakaryocytes and platelets. MPL, just like the JAK2 mutation, is active without the cytokine. So again, you don't need to enter the restaurant in order for this activity to occur. It's just going to happen by itself. So MPL is just another way to activate this pathway. It's similar to JAK2, and generally you wouldn't have both mutations in the same cell. They do the same thing. If you have a JAK2 mutation, only in rare cases would you also see a mutation in the MPL gene. And conversely, if you have a MPL mutation, you don't need a JAK2 mutation. You're doing the same thing into cells. Okay. So what's next? Well, this is complicated, but I think you'll understand it when I go through this. There are a lot of other mutations that have been identified, not just JAK2 and not just MPL. And again, you might hear some of these thrown about. You might not hear them outside of this meeting today. But they're important because these mutations, we think, work together with JAK2 to make this disease more complicated and more severe. And sometimes these mutations make you have a higher risk for developing, for progressing to leukemia, or these mutations might give you a higher risk of progressing to myelofibrosis, or they may decrease your survival. And so it's important to understand what these mutations are doing in cells and how they cooperate. So this is called the Circos plot, and it was developed, I've only started seeing these a few years ago. So what I'm showing you here are different patients that have different constellations of mutations. And so if you focus first on this part of it where it's written, where JAK2 is written, so this is the bottom right of the panel, so down here. These are patients that have JAK2 mutations. And what this plot is showing you is what other mutations are seen in those patients. What are the more common second mutations that you see? And so you can see that there's this very large red connector here. The red connector is showing you that when you have a JAK2 mutation, or these patients with a JAK2 mutation, also have a mutation in a gene called ASXL1. Okay? And don't worry about the names for now. I'll try to explain what these are doing in a moment. JAK2 individuals can also have this purple line, and this, I'm not colorblind, but my children are. So they also have mutations in other genes, such as TET2, or a protein called SRSF2, and I'll try to simplify what that is doing. So again, why am I showing this plot? It just illustrates the complexity of this disease, that you can have a JAK2 mutation, but then you also have something else. You have mutations in AXL1, for example. On the bottom right here are the percentages of the cases that have various mutations. It just tells you what's the incidence, how common are these? Should we be interested in studying those mutations? And the answer is yes, of course. That the top mutation here in JAK2, this is in myelofibrosis. It's about 50%, 59% in this cohort. This AXXL1, which you may not have heard of, but you should know because it's so common, is seen in about 20 some odd percent of these individuals. And then the next common one is something called TET2. Okay. So again, somewhat of a complicated diagram, but I want you to, just the take-home message from this is that in addition to a JAK2 mutation, there are other mutations that are seen, and they're in varying percentages. again, why are these mutations important? So what this graph illustrates here is when you look at a population of individuals that have different risk groups. So this is the IPSS scoring system, which you may or may not have heard about. It takes into account age, peripheral blood counts, and whether you have blast cells in the blood. And the IPSS scoring system goes from a low risk group to intermediate risk groups to a high risk group. And the high risk group is the group that has the greater predilection to developing leukemia or a much lower survival than the other groups. And so it's very important to understand what are the changes that give rise to this more intense or this higher risk disease. And so these little bar graphs are showing you what percentage of cases have mutations. And you can see that the JAK2 mutation, the percentage of cases with the JAK2 mutation is fairly common or consistent among these different groups. So JAK2 doesn't, just by having a JAK2 mutation, doesn't tell you what risk group you fall into. In contrast, the second one on the list is this AXXL1 gene that I'm harping on. This gene is much more commonly mutated in the high-risk group. So if you have this mutation in the presence of JAK2, it does suggest that you have a higher risk of transformation or a higher risk of dying sooner. And so we want to understand whether you have these mutations. So I think it's important for these genes to be sequenced in in your samples. The other ones here are not quite as prevalent or don't tell you all that much about the disease, except one other one. I think it's called SRSF2. This is a gene that it's called a splicing gene. I'll tell you what splicing is in a moment. And so, again, just underscoring that by knowing what these mutations are, we can be better positioned to understand what the prognosis would be. So, for these mutations, are there more mutations that we don't understand? And the answer is absolutely. We don't know everything about the disease. As I mentioned, for myelofibrosis, not everybody has a JAK2 mutation. Not everybody has a MPL mutation. There are clearly other genes that are mutated. And I suspect that these mutations do the same thing as JAK2. They activate this pathway. They turn on these important genes and pathways, STATs, PI3 kinase. And so we need to understand this. And the best way, I think, to get to this is by something called next-generation sequencing or DNA sequencing. That is where we can take samples of individuals and sequence all the genes. And that will then tell us what are some of the other mutations that are present. People are focusing on myelofibrosis without JAK2 mutations because what we really want to understand is how do you get myelofibrosis if you don't have a JAK mutation? And I think there'll be some important results coming out later this year. I don't know if you know, we have an annual American Society of Hematology conference. It's in the first week in December. And at that meeting is when we get to hear the most exciting advances in hematology research. We'll almost certainly hear about some new mutations. We'll also get an update, as you'll hear some today, about clinical trials that are ongoing for various JAK inhibitors or some of these other pathway inhibitors. So what are these mutations doing? And as I mentioned, I'm going to try to give you a quick overview of some of these other genes. And some of this might be a little complex, but I'm going to, again, do my best to make it simple. What do these mutations mean? Well, they affect signaling. Signaling is the first pathway we think about. This is what I showed you on one of the earlier slides, where you go to the restaurant and you signal to the waiter in the kitchen what you want. Those include mutations in these genes. Again, you don't have to memorize these, but JAK2, MPL, CBL, and LINK. Epigenetics is a term that I don't know if you may have heard this bantered around. This is important because many of the mutations that we see in this disease fall in this category. And this includes this important gene, AXXL1. So since that gene is associated with a poorer prognosis, we really want to understand how it's working. And so that we need to understand this process called epigenetics. And then finally, there's a pathway called RNA splicing, which I worked on as a graduate student, so it has a special place in my heart, which is why I have to tell you about it today. And that has this mutation in SRSF2. And this is the other mutation that gives rise to the worst prognosis. So it's important to understand what that mutation is doing as well. Okay, so what is epigenetics? Okay, so how would I explain this to you? Well, DNA, you probably have heard it's in a double helix. But this helix has to be bundled up very tightly in order for it to fit in your cell. So we have billions of base pairs. How do we get that into the small nucleus of a cell? Well, we wrap it up. So DNA is wrapped up first in beads on a string, we call this, where they wrap around proteins that are called histones. And histones are important epigenetic proteins or players. So it wraps around that, and then it subsequently wraps, they interact with each other, these different groups, these different beads on a string, and then they wrap like a phone cord to make it even tighter wound. And then eventually you make what are called chromosomes, and you've probably heard about chromosomes, of course, because chromosomal changes are associated with some of these diseases. So what is epigenetics? Well, epigenetics is the way that this is wrapped up. Epigenetic changes can dictate how tightly DNA is wound. And there are some genes, some important epigenetic factors. Oops, I always hit the wrong button. Epigenetic factors that modify or change histone tails. They modify what are called methyl groups. These, again, when you change these proteins or change these modifications, you change the way that DNA is organized. And by changing the way DNA is wound, you can then change gene expression. If it's bound too tightly, the cell can't turn on certain genes. If it's bound loosely, then you have a better chance of turning on these genes. And many of these genes then are disease genes. So if you have changes in epigenetic pathways, you can activate disease pathways. So the most common and most important gene that regulates this process is AXXL1. Okay, I keep promising that I'm going to tell you about splicing. And so what is splicing? Well, so splicing is like in the old days where you had a VHS cassette, let's say, or you have a movie on film, and you want to take out a scene. What you need to do is cut parts of that film, remove the part that you don't want, and then essentially tape the rest of it together. That's what splicing is. And so in genes, we have regions that are called exons. These are these colored, these are these colored boxes, and they need to come together in a certain pattern in order to make a normal protein. And so on the left example is a normal process where you get connection between the red, the yellow, and the blue. And so that makes this protein down there that has red, yellow, and blue. And that protein is a normal protein. It works the way it should. If you have changes in splicing, you change the way you the way you put this tape back together. And so on the right panel is shown if I'm going to fail this test here. Instead, you glue the red, the green, and the blue together, and you make what we call a mutant protein. So by changing splicing, you can change the way that cells grow. And this is important, again, because there's a gene called SRSF2 that's mutated in the worst cases. So we want to understand this process from a basic standpoint, and then to discover, can we change the way this happens, maybe with drugs? Okay, so now let's go to stem cells, and hopefully I'm doing okay on time. So what are stem cells? Well, stem cells are cells that have two properties. They can self-renew and give rise to the new stem cells, but they're also multipotent. That is, they give rise to multiple other cells in your body. And so the yellow cell is the stem cell, and when it divides, it gives rise to another yellow cell, another stem cell. But it can also give rise to other cells. These cells are of other colors, and these can be red blood cells, they can be platelets. But you need this stem cell to persist throughout your lifetime in order to keep your blood cells going. So we need these hematopoietic stem cells. So these are normal cells. And where do we see stem cells? Well, the most common stem cell you hear about is embryonic stem cell, or ES cells. That's not what I'm talking about here. I'm talking about cells that are present in adults. And we have a limited ability to regenerate certain tissues. We certainly can regenerate blood. And so blood uses these hematopoietic stem cells. And where do they live? They live in your bone marrow. But we have other stem cells, I won't go through all of them, but cells that can regenerate cells in the intestine, stem cells that regenerate hair, and other parts of your body. We also have something called mesenchymal stem cells. These are cells that live in the bone marrow, and they can give rise to a variety of tissues. And these are important potentially in fibrosis, because these cells, even though they're derived from bone marrow, they give rise to a variety of tissue types, including fat cells, adipocytes, chondrocytes, and myocytes, and these cells might be important in the fibrotic process. And I mentioned hematopoietic stem cells. These are cells that have self-renewing activity, and they give rise to all the blood cells in your body throughout time. And just simply showing a stem cell can give rise to red blood cells, but also white blood cells. Why am I harping on stem cells? Because there's a difference between normal stem cells and diseased stem cells. The normal stem cells create, as I mentioned, two daughter cells. One of those is another stem cell, and one is a committed progenitor cell, a red blood cell. that can't divide anymore. So you have a limited ability, not limited, but you have an ability to make normal cells from that. Leukemia or cancer stem cells are different. These are stem cells that keep the tumor growing. And so they divide to give rise to 2 daughter cells. And one of those is another cancer stem cell. So it allows the tumor to keep growing. And the other cell is, the other part of it, it can give rise to tumor cells, mature tumor cells that are in your body. Okay? Those tumor cells don't tend to proliferate, don't tend to keep growing. Tumors are composed of what we call heterogeneous cells, so a variety of different cell types that reside, in which reside a small population of these cancer stem cells. And just the purpose of this slide is just to say, well, how do we study stem cells? Well, normally you study cells by looking at bone marrow transplants, but you can also assess leukemia stem cells by bone marrow transplants. This is not the, this is not the final presentation, unfortunately, so I'm a little off here. Yeah, that's okay. Sorry about that. Yeah, I cut out some slides that I thought were a little too much, and so those are still here, unfortunately. This is a schema, though, that tells us how do we want to target cancer. The way that we traditionally target cancer is on the top panel, where we use conventional chemotherapies that kill off only these mature tumor cells. And so the green cells here are these tumor stem cells. They don't care about chemotherapy. They tend to be resistant. And so they stay in the body. So no matter, so you use an inhibitor, it might kill off the bulk of the tumor and you might feel a lot better, but there's an underlying persistence of these tumor stem cells that we'd like to get rid of. And so we want to develop what we call targeted therapy. We want to develop agents that will kill off both the tumor cells, bulk tumor cells, and the stem cell population. And so the question is, can we optimize therapy to target both cancer stem cells and the bulk of the tumor cells? And again, I'm going to have to skip through a couple slides. So these LSCs, or leukemia stem cells, are cells that allow a malignancy to persist in individuals. These cells tend to be resistant to current chemotherapies. But new data from mouse models, at least, suggests that interferons can provide some eradication of the tumor stem cell population. So I'm not a clinician, so I won't, I'm sure Dr. Silver will talk a little bit about interferons, but at least in animal models, it seems that they may be very effective at affecting both cells types. So what about, in the last few minutes, what about fibrosis? So, and you may have heard this term called the niche. The niche is part of your bone marrow where these cells live. What is a niche? And so to simplify that, I just took a few pictures from the internet. The niche is essentially, so you think about it as your bed. Very comfortable, very warm. And in particular, the hamburger, I think, looks really comfortable, doesn't it? That's not my bed. But each of these then, this forms the niche. This is where your bone marrow cells like to live. Your blood stem cells need to be in this warm environment. What happens when you have fibrosis? Well, instead of this normal, this comfortable bed, you have this bed of nails, or you have a bed of twigs. This guy seems pretty comfortable here, but I think in general, people are not happy sleeping on that. And neither are your blood cells. The hematopoietic cells don't need to be in that environment. And so in fibrosis, what often happens, or what does happen, is that the hematopoietic progenitor cells move out of the bone marrow and go into circulation, or can end up in other organs in the spleen commonly. And so we want to understand, is there a way to reverse this process? Can we take these bed of nails and convert it back to a nice bed, a comfortable bed? And those drugs are still I would argue, far away from the clinic, and are an important avenue of research both in academic labs and in drug companies. Why is the niche important again? Well, it provides nutrients and signals to hematopoietic stem cells and other bone marrow cells that provide for growth. And an abnormal niche is associated with blood malignancies and other blood disorders. And I have to, just a couple more minutes. So what causes fibrosis? Well, there's two components. One is what I call the inducing cell. It's something that has a mutation that then changes the environment. So cells with the JAK2 or MPL mutation that secretes factors to remodel cells that lead to fibrosis. The receiving cell is the cell that responds to these abnormal signals. This is the cell that lays down these fibers. And so we want to be able to target both of these different processes, or at least one of them. And these inducing cells can be, we think, megakaryocytes. They can be other kind of myeloid cells or other bone marrow cells. I guess the point here is we don't actually know what they are. The receiving cells are other cells in your bone marrow, not the blood cells. They can be these multipotent, what we call stromal cells, or bone-forming cells, osteoblast lineages. Can also be adipocytes. So there's a study ongoing now to ask, are fat cells in your bone marrow, are those the ones that lay down fibers? Or we think a variety of other types. Again, we don't really know enough about that. And the last two slides are a little more complicated, but illustrates the state of the art of the field. Two more slides. So this is the normal niche where your hematopoietic stem cell lives, and it's happy. It can give rise to myeloid cells and other cells. In the environment here, it's kind of simple. What happens when you have fibrosis? Well, just the take-home message here is it's a mess. Things are much more complicated. These hematopoietic stem cells don't like this environment, but the cells that do like the environment to some degree are these leukemia stem cells or malignant tumor cells. And in this publication that recently came out, there's a proposal that myeloid cells are the signals and that the receiving cells are what are called osteoblasts, and that these promote fibrosis. And so the insight here is that we might be able to target this by targeting these particular initiating cells or these receiving cells. And so finally, to summarize, there have been several exciting advances in our understanding of the molecular basis of this disease, and also to the limitations of JAK therapy. And again, I'll let other people today speak about that. There have been recent developments in new therapies to be used in combination with JAK inhibitors, which I didn't have time to tell you about. But we think that some of these new drugs might be able to cooperate with JAK inhibitors to help cure the disease. And finally, there have been many advances in understanding the bone marrow and splenic niches and the possible causes of fibrosis that we hope then will allow us to better target that